So, good evening, guys. We'll start with the today's class. So, today we'll be starting with discussion week 7 of the biochemistry course of from NPTEL. So, let's start. So, this is the first question for today. Which of the following occurs during the oxidation of glyceraldehyde 3-phosphate into 1,3-biphosphoglycerate in glycolysis? So, what can be the correct answer? Okay. So we have two A and B. Okay. Any more answers? B and C. Okay. Okay. Any more answers? Okay. So the correct answer is A and D. Okay. So this is the uh, procedure that happens during the conversion of glyceraldehyde 3-phosphate to 1,3-biphosphoglycerate, and this is the mechanism of the reaction. So what happens? This is the glyceraldehyde 3-phosphate dehydrogenase that converts the glyceraldehyde 3-phosphate to 1,3-biphosphoglycerate. Okay. So now what happens? NAD is present as a cofactor here, and it has two groups, that is cysteine and histidine, that are basically involved in the reaction. So these are the basically forms the active site of the molecule. So what happens first? When the glyceraldehyde 3-phosphate enters the enzyme, it forms a complex. So this is a thiohemiacetal intermediate that is formed because it is formed between the sulfur group. So that's why it is a thiohemiacetal. Okay. So a thiohemiacetal intermediate is formed. Then what happens? This is the thioacetal intermediate basically. Okay. So okay, wait, I am just explaining the reaction mechanism first, then we'll see the options. Okay, so first the thiohemiacetal intermediate is formed. Okay, and then the oxidation of thioester intermediate is basically done by the NAD plus. Okay, so that is done by the NAD plus. So now NADH is actually released, and the again now we have to release the product. So what happens? The pi, that is inorganic phosphate, actually enters into the medium, and then it is formed, the second phosphate is added to the product, and the 1,3 biphosphoglycerate is released from the enzyme. Okay, and here the NAD plus is actually uh, replaced by the NADH, because NADH is released from the enzyme. So NAD plus is replaced by the NADH is replaced by the NAD plus. Okay, so this is the full reaction mechanism that takes place from conversion of glyceraldehyde 3 phosphate to 1,3 biphosphoglycerate. Okay, now let's see the options. So first, what happens? Addition of a phosphoryl group. So yes, because here inorganic phosphate is added, that's why it is 1,3 biphosphoglycerate. Okay, then covalent interaction between a sulfhydryl hydrogen and a imidazole nitrogen and an enzyme's active site. So no, this is not a covalent interaction. Okay, so this is not a covalent interaction. Basically, this might be a non-covalent interaction that happens between the cysteine and histidine. Okay, so this option is false. An aldehyde is converted to a free carboxylic acid, a free carboxylic group. No, the aldehyde is basically converted to an is attached to a phospho group. So this is not a carboxylic group basically. Okay, so this is also a wrong option. Then a thioacetyl intermediate is oxidized by NAD plus. Okay, so what happens? A thiohemiacetal would be the correct option. Okay, thiohemiacetal would be the intermediate that is formed, and that is actually being oxidized by the NAD plus. Okay, so yes, this is also a correct answer. Two molecules of NAD plus are oxidized per molecule of glyceraldehyde 3 phosphate. No, per one molecule, only one NAD plus is oxidized. Okay, not two. So for two molecules of glyceraldehyde 3 phosphate, two molecules of NAD would be required. Okay, so this option is also false. Then there is no net gain of 2 electrons per molecule of glucose conserved in the form of NADH. Okay, so there is net gain of 2 electrons per molecule of glucose conserved in the form of NADH. No, this is also false, because only one electron is added. Sorry, for one molecule of glucose, there would be two NADH will be formed. So that means 4 electrons would be required, because for one NADH, 2 electrons are required. Okay, so this is also a false option. So the correct ones are A and D. Here it would be thiohemiacetal, not thioacetal. Okay, so is the explanation? Okay, any doubts in this question? Okay, so let's move on to next question. Which of the following statements are incorrect about the part yes is not correct? Okay, so if we see, when NAD plus is actually converted to NADH, okay, two electrons are basically involved in the reaction. Okay, and for one glucose molecule, two glyceraldehyde 3 phosphate are formed. Okay, so that would require uh, two NADH molecules. Okay, so that means two NADH are formed. That means 4 electrons are actually conserved in that case, not two. because here per glucose molecule is like asked. Okay, yes, someone raised the hand. Okay, so thioacetyl. So actually, acetyl is a group that is formed by the aldehyde group. Okay, so aldehyde, when it reacts with any of the other molecules, it forms a acetyl group. Okay, so as you can see here, this is the aldehyde group from the glyceraldehyde, and it is forming a acetyl group. Okay, so what happens when this aldehyde group is attached to any other molecule? When it's actually in reaction, it forms a acetyl group. So here a thioacetyl, that means a thio means whenever there is a thio, that means a sulfur group is involved in the reaction. Okay, so this thio and the aldehyde group, when it forms a reaction, it forms a thiohemiacetal. Okay, and why it's hemi? Oh, so basically, I don't know about this. I'll let you know after checking. Okay, and what was the other question? The E option, right? So, if we can see, 1 glyceraldehyde 3 phosphate from here is actually it's converted to 1,3 biphosphoglycerate. It utilizes one NAD when it NAD plus molecule, and it releases one NADH. Okay, and E option says that two molecules of NAD plus are oxidized per molecule of glyceraldehyde 3 phosphate. So only one molecule is actually oxidized here, not two. That's why it's false. Okay, is it clear now? Hmm. Okay, so let's move on to the next question. Which of the following statements are incorrect about the part of glycolytic pathway up to the formation of 1,3 biphosphoglycerate? So, what can be the correct answer? Okay, A and B. So, we have got first answer. Just make sure that you are telling the incorrect statements. C, D, and E. Okay, and D and E. Okay, so let's see the answer. So it's A, C, E. Okay. So let's check the options first. Yes, let's check the options. Okay, for one molecule of glucose, there is two molecules of ATP are formed. Okay, so if we see from one glucose molecule, okay, one ATP is actually utilized for the conversion of glucose to glucose 6 phosphate, and two molecules of ATP are formed. Okay, so that is from the phosphoenol pyruvate to pyruvate. Okay, so in that case, the net ATP that is formed from this is 1 ATP. Okay, here we are not counting the number of ATPs from the NADH molecule. Okay, so the net ATP that is formed from one glucose molecule is 1 ATP. Okay, so this option is false. Fine. Then for one molecule of glucose, net two molecules of NADH are formed. Yes. So from the last question also we have discussed, from glyceraldehyde 3-phosphate to 1,3-biphosphoglycerate, one NADH molecule is released. Okay. And because from one glucose molecule, two molecules of glyceraldehyde 3-phosphate are formed. So that means two molecules of NADH will be released in the whole process. Okay, so this option is actually correct. Then option C, one substrate level phosphorylation reaction occurs in the full process. No, this is false. Okay, just 1 minute. Okay, so substrate level phosphorylation is basically a reaction in which the substrates are directly involved in formation of ATP. Okay, so there are two steps where the substrate level phosphorylation actually occurs. First step is the conversion of glucose to the glucose 6 phosphate. Okay, because here the directly ATP is involved in transferring the phosphate group, and the second reaction is from phosphoenol pyruvate to pyruvate. Here, two molecules of ATP are released. Okay, so these two are the substrate level phosphorylation reactions that happen in glycolysis. Okay, so C option is false. Then a total of two protons are consumed per NADH molecule formed. Okay, so what happens generally, the way of writing is NADH2 or NADH plus H plus is the correct way of writing NADH. Okay, so how we write it? It's actually two protons that are added to one NADH molecules. Okay, so that's why this is a correct answer. Then a total of two NAD plus molecules are oxidized. Okay, so uh, I think this would be the correct answer, because from we are getting as two NADH molecules, but two NAD plus molecules are also added up. So these two NAD plus molecules are oxidized in the whole process. So this is a correct answer. Okay, so this is a misprint. Only A and C are incorrect answers. Okay, is the explanation clear? Okay, here is a correction. Okay, So as we write this NADH plus H plus, only one proton is added here. Okay, so no two protons, this would be one proton. So instead of E, this would be D. That would be the incorrect answer. So the total of incorrect would be A, C, and D. So any doubts in this question? If not, then we'll move on to the next question. Which of the following enzymes do not function in the payoff phase of glycolysis? So what can be the correct answer? Okay, C, E, and F. Okay, any more answers? C and E. Okay, G also. Okay, okay. So the correct answer is C, E, F, and G. Okay, so here is the explanation. So first, when we enter the glycolysis, so there are two basic phases that are being involved. First is the preparatory phase, in which glucose is actually being prepared to glyceraldehyde 3 phosphate. Further, when the payoff phase begins, when glyceraldehyde 3 phosphate is converted to pyruvate, and here the actual uh, derivation of energy starts. Okay, so in the first phase, the ATP is actually being involved. Okay, and uh, in the second phase, the energy is being released. Okay, so this means that is the payoff phase. Okay, so we'll check what are the enzymes that are involved in the preparatory phase. So first, when glucose is converted to glucose 6 phosphate, it takes place with the help of hexokinase. Then glucose 6 phosphate is converted to fructose 6 phosphate. This is a phosphohexose isomerase. Okay. Then when fructose 6 phosphate is converted to fructose 1,6 biphosphate, this is phosphofructokinase 1. Okay, then fructose 1,6 phosphate is broken down into glyceraldehyde 3 phosphate and dihydroxyacetone phosphate. This is done by the enzyme aldolase. Okay, now this dihydroxyacetone phosphate is actually isomerized to glyceraldehyde 3 phosphate. Both are isomers. So this is triose phosphate isomerase. So these are the five enzymes that are involved in the preparatory phase basically. Okay. Now, in the after this, the payoff phase starts, and glyceraldehyde 3-phosphate is converted to 1,3-biphosphoglycerate by the enzyme glyceraldehyde 3-phosphate dehydrogenase. Then further, 1,3-biphosphoglycerate is converted to 3-phosphoglycerate by phosphoglycerate kinase. Okay. Then 3-phosphoglycerate is isomerized to 2-phosphoglycerate by phosphoglycerate mutase, and then 2-phosphoglycerate is converted to phosphoenol pyruvate by enolase. Then phosphoenol pyruvate is actually finally converted to pyruvate by the enzyme pyruvate kinase. Okay, so let's see the options. So enolase is involved in the payoff phase. Pyruvate kinase is also involved in the payoff phase. Aldolase is involved in the this step. Okay, so it's in the preparatory phase. So this is not involved in the payoff phase. Then phosphoglycerate mutase is actually a isomerized enzyme. So this is involved in the payoff phase. Phosphofructose kinase 1 is the enzyme involved in the formation of fructose 1,6 biphosphate. So this is also involved in the preparatory phase. Then glyceraldehyde 2 phosphate dehydrogenase is also okay. So this enzyme is not involved in the glycolysis at all. Okay. So this is also the correct answer because it is not involved in glycolysis. Then glyceraldehyde 3-phosphate hydrolase is also an enzyme that is not involved in the glycolysis. Okay. Then phosphoglycerate kinase is the enzyme that is involved in the payoff phase. So yes, this is also involved. So the correct answer is option C, E, F, and G. Okay. So any doubts in the explanation? Okay, if no doubts, then we'll move on to the next question. So identify the correct statements from the given following statements. So what can be the correct answer? Okay. Any more answers? D. Okay. Make sure you find out the correct options only. Okay. So D, E, and F. Mhm. Okay. So let's see the answers. So only the correct option is E. Okay. So let's see the options. Conversion of glucose to pyruvate gives 3 ATP molecules. No, this is a false statement. If we see 2 ATP, 4 ATP are formed and 2 ATP are actually consumed during the reaction. So the net gain of ATP is 2 ATP. Okay, so this is a false option. Then the fermentation is the process of generating energy from proteins without the use of oxygen. So no, it actually utilizes it can utilize glucose or basically carbohydrates. Okay, although it is the use without the use of oxygen, that means it's an anaerobic process, but it is not used for deriving energy from the proteins. Okay, so this is also not a correct answer. Pyruvate decarboxylase is present in the human muscle cells. No, it is present in every cell because pyruvate decarboxylase is the enzyme that is involved in the conversion of pyruvate to acetyl coenzyme A. Okay, so this is also an incorrect option. Then net gain of ATP from the conversion of phosphoenol pyruvate to pyruvate per molecule of pyruvate is 2. Okay, so this is also a false option. Why? Because when one molecule of phosphoenol pyruvate is converted to pyruvate, so what happens? One molecule of ATP is released. Okay, so for one molecule of pyruvate to be formed, one ATP molecule will be released. Fine. So if we see two molecules of PEP will form two molecules of pyruvate, then two molecules of ATP will be released. So for one molecule of pyruvate, one molecule of ATP will be released. Okay. So this is also an incorrect statement. Then thiamine pyrophosphate carries an active aldehyde group. Yes, This is a correct statement. Okay, so this is in the figure as we have seen. This is a thymine pyrophosphate molecule, and through this group, okay, the active aldehyde molecule is actually attached. Okay, so this is a correct answer. Then the carbon 2 in the thiazolium ring is in TPP, readily dissociates a proton and forms a carbocation, which readily reacts with the carbonyl carbon. Okay, so here the carbon is actually releasing the H plus. This is the second carbon that is actually attached to the active aldehyde, and then it is attached to the carbonyl group. Okay, so yes, this is a correct answer. Okay, so here F would also be the correct answer. Then thiamine deficiency causes pellagra. No, thiamine deficiency causes beriberi. Okay, then lowering of glucose level in the blood is called hyperglycemia. No, it's called hypoglycemia. Okay, so the correct answer is option number E and F. Okay, any doubts in this question? Okay, so as you know, that one molecule of phosphoenol pyruvate actually yields pyruvate, right? So one molecule of phosphoenol pyruvate yields one molecule of pyruvate. Now it releases one molecule of ATP in this reaction. Okay. Now, for one glucose molecule, if we consider, this would be two molecules of phosphoenol pyruvate and two molecules of pyruvate that are formed with the release of two ATP molecules. Now, in the option, the net gain of ATP from the conversion of phosphoenol pyruvate to pyruvate per molecule of pyruvate. So that means one molecule of pyruvate is formed. In that case, the net gain of ATP is 2. No, the net gain of ATP is 1 when one pyruvate molecule is involved in the reaction. Okay, so that's why this option is incorrect. Now the explanation is fine. Mm, the reaction of TPP. Option F, you are saying. Yes, F option is correct because in the case, yeah, that is misprint in the answer. So F option is correct. Okay. So in this case, what happens? The carbon 2 is release this H plus and it forms a carbocation. So this carbocation is actually an active form. So it reacts with the carbonyl group of the acetaldehyde. Okay. So this is a correct answer. F. Okay, shall we move forward? Okay, so, okay, so as you can see in this image, okay, this is the TPP molecule that is shown. Okay, this is a thiazolium ring, and here the second carbon is the carbon through which an active aldehyde is attached. Okay, so the E option says that the thiamine pyrophosphate carries an active aldehyde groups. Okay, so yes, this is a correct answer. Now, moving to option F, as you can see, this is the second carbon. How does it attach to the active acetaldehyde? This is the explanation. Okay, so first, what happens? This hydrogen from this second carbon is released. So this is actually the attached aldehyde group. Okay, so this carbon is the carbonyl carbon. After attaching to the thiazolium ring, it has formed the hydroxyl and this H plus ion. Okay, so the actual acetaldehyde molecule is like this.
Sir,
Sir, uh,
means
means
thymine
thymine
pyrophosphate
pyrophosphate
is
is
reacting
reacting
with
with
hydroxylethyl
hydroxylethyl
thymine
thymine
pyrophosphate,
pyrophosphate,
or
or
the thymine
thymine
pyrophosphate
pyrophosphate
after
after
reacting
reacting
with
with
acetaldehyde
acetaldehyde
is
is
giving
giving
hydroxyethyl
hydroxyethyl
thymine
thymine
pyrophosphate.
pyrophosphate. So after reacting with acetaldehyde, it is giving hydroxyethyl thymine pyrophosphate.
Okay,
Okay.
sir.
Hmm. Okay, so the explanation is clear.
Okay,
Okay.
sir.
Okay,
Yes,
so
sir.
let's move on to the next question. So, which of the following pathways can give rise to glucose? So, what can be the correct answer? Okay, A and C. Okay, so you meant except for E, all are correct. Okay. Any more answers? Okay. So the correct answer is option F. Okay. So let's see how. So the first option, lactate to pyruvate, then passes through TCA and to give glucose 6 phosphate. Okay. So yes, this is the correct pathway. Here in the orange one, it can give to rise to glucose. Okay. Glucose 6 phosphate basically. So the lactate is converted to pyruvate, and then pyruvate can enter through citric acid cycle, or it can directly enter through gluconeogenesis. Okay, so this is the correct pathway. Then carbon dioxide fixation by plants give rise to 3-phosphoglycerate, which then converts to glucose 6-phosphate. So yes, it gives rise to 3-phosphoglycerate. Okay, and then it can directly enter through gluconeogenesis to form glucose. Then hydrolysis of triacyl glycerol to glycerol and then to glucose 6 phosphate. Okay, so triacyl glycerols can give rise to glycerol, and glycerol can directly be converted to glyceraldehyde 3 phosphate. Okay, which can directly enter the gluconeogenesis pathway. So it can also give rise to glucose 6 phosphate. Then glucogenic amino acids through the TCA cycle to glucose 6 phosphate. So yes, glucogenic amino acids are those amino acids basically that are formed from glucose. Okay, or that are formed from the TCA cycle. So these amino acids can directly form to the citric acid cycle and can give rise to glucose 6 phosphate. So yes, this is also correct. Then free fatty acids can also give rise to glucose through glucose 6 phosphate. Okay, so what happens here? The free fatty acids basically are in the form of actually known pathway is involved in the formation of glucose. Okay, so that's why this is a very trivial thing. Sorry, that is an ongoing research thing, but there are papers that actually suggest that the free fatty acids also give rise to glucose. Okay, so this answer is not really correct, but it's also correct in the with the growing research. So except for E, for now, all the options, other options are correct. Okay, so it would not be F basically, but it would be from A to D. Okay, so any doubts in this question? Okay, so in one of the pathways, what happens? The free fatty acids that we are talking about. So by beta oxidation, they can actually be yield acetyl coenzyme A. So this acetyl coenzyme A can actually involved in the citric acid cycle and can give rise to glucose. Okay, so yes, this can also be the case where free fatty acids can actually be involved in the production of glucose. Okay, so hence all of the options are correct. So, any doubts in this question? Okay, if not, then we'll move on to the next question. Which of the following statements are true about the tricarboxylic acid cycle? So, what can be the correct answer? Okay, A and E. Okay, any other answers? You have to identify the true statements. Okay, C and F also. Okay, any more answers? Okay, this is not correct. Okay, so the correct answer is C, D, and E. Okay, so let's see the options. This is basically the okay. So acetyl coenzyme A is reduced in the TCA cycle. No, acetyl coenzyme A is basically oxidized in the TCA cycle. Okay, why? Because it loses its electrons to NAD plus and FAD plus. Okay, so it's basically oxidized, not reduced. Then glucose is the only source for acetyl coenzyme A. No, this is also not correct because just now I told you that free fatty acids, while oxidizing, it also produces acetyl coenzyme A. Okay, so this is also not correct. Then oxidation here means electrons flow from acetyl coenzyme A to NAD plus and FAD. Yes, this is correct. Okay, we are talking about the oxidation of acetyl coenzyme in the TCA cycle. So the electron flow occurs from the acetyl coenzyme to these molecules, that is NAD plus and FAD. Okay, so this is correct. Then electrons are transferred from NADH and FADH to oxygen via the oxidative phosphorylation. Yes, this is also correct. So in the mitochondria, what happens? These reduced forms of NADH and FADH2 are formed, and then these transfers the electrons to the oxygen, forming the metabolic water. So this process is called as the oxidative phosphorylation, and this happens in mitochondria. So yes, this is also correct. Then electron transfer occurs in the mitochondria utilizing electrons from the TCA cycle. So basically, the electrons are involved with the help of NAD plus NADH and FADH2. So these enter the oxidative phosphorylation to transfer the electrons to the final oxygen molecule. Okay, so yes, this happens in the mitochondria utilizing electrons from the TCA cycle. Then the electron gradient is used to make NAD plus. No, this is false. Okay, because the electron gradient is finally maintained to form the ATP molecule and the metabolic water. Okay, not to form NAD plus. So the correct statements are C, D, and E. Okay. So, any doubts in this question? Okay. If not, then we'll move on to the next question. Which of the following are components of coenzyme A? So, what can be the correct answer? Okay. B. Any more answers? Okay. So, the correct answer is option B and D. Okay. So, pantothenic acid and adenine. So, this is the structure of the acetyl coenzyme A. So basically coenzyme A. So it has a adenine molecule, ribose 3 phosphate, ribose 3 prime phosphate. Okay, so this is the phosphate group that is attached to the ribose molecule, and then at the 5 prime end it has two other phosphate molecules that are attached to it. Okay, so this is basically 3 phosphoadenosine diphosphate. Okay, so this is the whole molecule that is being involved here. Then it has pantothenic acid, then it has beta mercaptoethylamine. Okay, and this HS group is the reactive thiol group from where the acetyl group is attached. Okay, so this is the acetyl group and it is attached to this beta mercaptoethylamine. Okay, so this forms the acetyl coenzyme A basically. Okay, so from the options that we have seen, the pantothenic acid and the adenine are the components of coenzyme A. Acetyl group is a component of the acetyl coenzyme. Okay, so the other components are not involved in the coenzyme A molecule. Okay, this must be diphosphate. Okay, this is triphosphate. This must be diphosphate. So that's why this is not a component of coenzyme A. Then beta mercaptoethanol, not ethanol, it must be ethanol amine. Okay, so this is also not involved. Then thymine pyrophosphate and acyl lipoate is also not involved in coenzyme A molecule. So the correct answer is option number B and option number D. Okay, so any doubts in this question? Okay, so let's move on to the next question. In TCA cycle, which of the following statements are incorrect? So, what can be the correct answer? You have to find the incorrect statements. Hmm. Yes, someone raised a hand. Okay, any more answers? B and C. Okay. A and G. Okay. Yes, someone raised a hand. You can ask the doubt. Okay. So the correct answer is A, G, and H. So these are the incorrect statements. Okay. So let's see. So this is a typical TCA cycle that we are seeing. So once the acetyl coenzyme molecules enters the cycle, it reacts with oxaloacetate and forms citrate. Okay, here one molecule of water is entered and the coenzyme molecules is released. Okay, now this takes place with the help of citrate synthase. Now, this citrate molecule is dehydrated. That means one molecule of water is removed, and the enzyme is aconitase, and it forms cis aconitate. And then, after adding another H2O molecule, the isocitrate is formed. Okay, here the enzyme is involved. So, this is basically a isomerization from citrate to isocitrate. Now, this isocitrate is converted to alpha ketoglutarate by releasing one molecule of CO2, and this also forms one molecule of NADH. So, this is isocitrate dehydrogenase. Okay, then uh, the alpha ketoglutarate converted to succinyl coenzyme A when another coenzyme A molecule is entered into the cycle and one molecule of CO2 is released. Okay, so this is basically another molecule of carbon dioxide is released. So this is a decarboxylation reaction, and this is done by alpha ketoglutarate dehydrogenase complex molecule. And here also one NADH molecule is formed. Okay, then the succinyl coenzyme A is converted to succinate by conversion of the GDP molecule to GTP. Okay, and the coenzyme A molecule is released. So this is succinyl coenzyme A synthetase. Then this succinate molecule is converted to fumarate by an enzyme called as succinate dehydrogenase, and this yields a molecule of FADH2. Okay, then this fumarate is actually converted to malate by the enzyme fumarase, and another molecule of water is involved in the reaction. Okay, then this malate is actually converted to oxaloacetate by the conversion by the enzyme malate dehydrogenase, and another molecule of NADH is formed. Okay, so this is the whole TCA cycle that we can see. Now we'll see the options. So the citrate to isocitrate conversion involves an enzyme isocitrate dehydrogenase false. The enzyme involved is aconitase. Okay, then fumarate to malate, it's fumarase. So yes, this is correct. Okay, because from fumarate to malate, fumer is involved. Then malate to oxaloacetate, malate dehydrogenase is involved. Yes, this is correct. So from malate to oxaloacetate, malate dehydrogenase is involved. Then from succinate to fumarate, FAD is converted to FADH2. Yes, this is also correct by the enzyme succinate dehydrogenase. Okay. Then succinyl coenzyme A to succinate, GDP is converted to GTP. So yes, this is correct. By the conversion of succinyl coenzyme A to succinate, GDP is converted to GTP. Then alpha ketoglutarate to succinyl coenzyme A, one CO2 molecule is released. So yes, this is also correct. From alpha ketoglutarate, the CO2 molecule is released, and this is converted into succinyl coenzyme. Then three NADH molecules are reduced per cycle. So three NADH molecules. So one from here, and one from here. Okay. So yes, this is a correct answer. Then net three H2O molecules are consumed per cycle. Okay. So let's see. One molecule is consumed here. Then one molecule is consumed here, and one molecule is consumed here, and one molecule is released here. So that means net two H2O molecules are consumed per cycle. Okay, three are actually consumed and one is released. So that means two water molecules are consumed. So this is a false answer. Then NAD plus is converted to NADH in the process. So yes, this is a correct answer. Okay. So the incorrect statements would be option A and option H. Okay. So G would not be an incorrect answer. Okay. So any doubts in this question? Okay, if no doubts, then we'll move on to the next question. Which of the following statements are correct? So, what can be the correct answer? Okay, any more answers? Okay, So the correct answer is A, C, and F. Okay. So A is correct, C is correct, and F is correct. Okay. So let's see. Delta G for the conversion of succinate to fumarate is 0. Okay. So yes, as we can see here, the succinate to fumarate is done by the enzyme succinate dehydrogenase, and the hydrogen molecule is actually transferred to the FAD. Okay, so both electrons and the H plus ion are transferred to FAD molecule to form FADH2. Okay, so this in this reaction the delta G naught is actually 0. Okay, so this is correct. Then succinate dehydrogenase is attached to the outer membrane of the mitochondria. No, it is attached to the inner membrane. Okay. From succinate to fumarate, the electron transfer is in the form of hydrogen. Yes, this is correct. Okay. Then, as we have seen now, these two hydrogen molecules are basically transferred to the FADH2 molecules. Okay. Fumarase is stereospecific and takes L-malate to form fumarate from in the reverse reaction. Okay. So, no, this is false. Okay. Because as we can see here, the fumarate is actually convert is no actually it takes d-malate to form fumarate. Okay, in the reverse reaction. So remember one thing, in carbohydrates we use only d form. Okay, not L form. Only for amino acids we use L form. Okay, so this is actually a false statement. Then the total number of ATP formed when glucose is completely oxidized to carbon dioxide is around 30 to 32. So this is also false. Okay, so what is actually the number of ATP molecules that are formed from one glucose molecule after the complete oxidation of glucose? Is it 36? Okay, you guys let me know the answer in the next class. Okay, so now moving on to the last option, intermediates in the TCA cycle serve as precursor in many other biosynthetic pathways. Yes, this is a correct answer. So the correct options are A, C, and F. Okay, any doubts in this question? Okay, if not, then we'll move on to the next question. Identify the incorrect statements in the following list. Any more answers? Okay. So the correct answer is option C, D, and E. Okay. So let's see. Pyruvate carboxylase is a regulatory enzyme that is activated by acetyl coenzyme A. Yes, this is a correct statement. So pyruvate carboxylase is an enzyme that converts pyruvate to oxaloacetate. Okay, it can directly convert from pyruvate to oxaloacetate. So this enzyme is actually activated by the acetyl coenzyme A. Okay. Then PEP carboxykinase is present in the heart and skeletal muscle. So PEP carboxykinase is the enzyme that is involved in gluconeogenesis, and it is predominantly present in the heart and skeletal muscles. Then PP carboxylase is present in the liver and kidney and is involved in gluconeogenesis. No, this is a false option. Then malic enzyme is limited to prokaryotes. No, this is also false because it is involved in eukaryotes also. Then biotin acts as a carrier to transport CO2 from one active site to another and on different enzymes. No, biotin is actually involved in it's actually acts as a carrier for transfer of the hydride ions. Okay, so it doesn't transfer carbon dioxide. The overall end product regulates the enzyme in the TCA cycle negatively. So yes, this is correct. Okay, because the TCA cycle is actually a regulated process. So all the end products, the overall end product, that means carbon dioxide, ATP, NADH, all these regulate the enzymes of the TCA cycle. Okay, so option C, D, and E are the incorrect answers. Okay, so any doubts in this question? Okay, so let's move on to the next question. Assume you discovered a mutant yeast whose glycolytic pathway was shorter because of the presence of a new enzyme catalyzing the reaction given below. Which of the following is likely to happen? So glyceraldehyde 3-phosphate plus hydrogen gives 3-phosphoglycerate and it yields NADH. Okay. So what can be the correct answer? Okay. Any more answers? Okay, so the correct answer is option A and option C. Okay, so let's see. So here it states that the process of glycolysis is a bit shorter because the step from glyceraldehyde 3 phosphate to 1,3 biphosphoglycerate to 2 phosphoglycerate and then to 3 phosphoglycerate. Okay, so these three steps are basically subsided by one enzyme. Okay, and here in this, only one NADH molecule is formed, and here only one ATP molecule is formed. Okay, so that means the NADH is already forming here. So there is not much happening in the glycolytic pathway by the removal of these three steps. Okay, so that means the mutation, sorry, the presence of this new enzyme will not as it is affect the glycolytic pathway. Okay, so the reaction or the yeast would be growing in the same same environment as it was before, or in the normal wild type case. Okay, so that means it cannot grow in the anaerobic conditions, and it will not have any observable effect in the aerobic conditions. So it can grow freely in the aerobic conditions. Okay, but it will not be able to grow in the anaerobic conditions. Okay, so options A and C are correct here. Okay, any doubts in this question? Okay, if not, then we'll move on to the next question. Deficiency of which of the following vitamins will affect the oxidation of glucose? So, what can be the correct answer? Okay, any more answers? Okay, okay. So, the correct answer is option A, B, and C. Okay, so riboflavin is involved with FAD. So FAD is basically involved with succinate dehydrogenase. Okay. Then thiamine is TPP, thiamine pyrophosphate. This is involved with pyruvate decarboxylase and alpha ketoglutarate dehydrogenase. Okay. Then niacin is NAD molecule, and that is involved with glyceraldehyde 3-phosphate dehydrogenase. Then this one, pyruvate decarboxylase also. Then we have isocitrate dehydrogenase, okay, and many more. Okay, so these three vitamins, if there is deficiency, then that would affect the oxidation of glucose. Okay, so any doubts in this question? Okay, if not, then we'll move on to the next question. Which of the following enzymes require Mg2 plus for their activity? So, what can be the correct answer? Okay, any more answers? Okay, so the correct answer is option A, B, and D. Okay, so all the enzymes that work for substrate level phosphorylation. Okay, so they actually require Mg2 plus. Okay, or the reactions that require ATP, either formation of ATP or utilization of ATP, those reactions also require Mg2 plus. Okay, so pyruvate carboxylase is conversion of pyruvate to oxaloacetate. So in that case, ATP molecule is involved. So this requires Mg2 plus. Then phosphoglycerate mutase is the enzyme that converts 2-phosphoglycerate to uh, yes, to 3-phosphoglycerate. So this also requires Mg2 plus. Then phosphoglycerate kinase, it requires Mg2 plus because it is the enzyme that converts 1,3-biphosphoglycerate to 3-phosphoglycerate. Okay, so it helps in the formation of ATP. So these three enzymes actually requires Mg2 plus. Okay, any doubts in this question? Okay, if not, then we'll move on to the next question. Which of the following processes will occur under both aerobic and anaerobic conditions in humans? So what can be the correct answer? Any more answers? Okay. So what happens if we see glycolysis first? Okay. Glucose is converted to pyruvate, two molecules of pyruvate. Now, this pyruvate can actually go to either lactate formation or ethanol formation through fermentation, or it can go to acetyl coenzyme A and then further formation of carbon dioxide and water. This is through TCA cycle. Okay, so this part is the aerobic reaction, and this part is the anaerobic reaction. Okay, so when glucose is actually converted to pyruvate, in that case, it is an essential reaction for both anaerobic as well as aerobic conditions. Okay, so even if an organism is growing in absence of oxygen and it can actually do fermentation, then glycolysis will happen and fermentation will happen. And for aerobic organisms, fermentation will not happen. Okay, but the glucose will directly be involved in the TCA cycle. Okay, so this is a common step, glycolysis, that happens for both anaerobic and aerobic conditions. Okay, so the correct answer is option number A. Okay, is the explanation fine? Okay, so let's move on to the next question. The structure of vitamin B7 is shown below. The functioning of which of the following enzymes is likely to be affected by vitamin B7 deficiency? Okay, so the correct answer is option number A, that is pyruvate carboxylase. So vitamin B7 is the other name for biotin. Okay, and biotin is involved in the enzyme called as pyruvate carboxylase that converts pyruvate to oxaloacetate to maintain the homeostasis. Okay, so the deficiency of biotin will directly affect pyruvate carboxylase. The other enzymes are basically involved with pyruvate decarboxylase. So let's move on to the next question. The energy cost of transforming 10 glucose molecules to pyruvate via glycolysis and back to glucose via gluconeogenesis is dash ATP equivalents. So you have to calculate the number of ATP molecules that are consumed for converting 10 glucose molecules to pyruvate and then back to glucose via gluconeogenesis. Hmm, 80. Okay. Any more answers? No, you have to calculate the net ATP molecules. Okay. So in glycolysis, if we see one glucose molecules form two pyruvate molecules that require addition of two ATP molecules and release of four ATP molecules. Right. But in gluconeogenesis, when the reverse happens, so 6 ATP molecules are actually conserved for forming glucose molecule. Okay. So if we see for 10 glucose molecules in gluconeogenesis, 60 ATP would be required. Okay. And if we talk about glycolysis, 20 ATP would be required. Okay. So this would yield 80 ATP. And 4 ATP are formed in glycolysis. Okay, so this would yield 40 ATP. From one glucose molecules 4, so 10 molecules of glucose will yield 40. So the net would be 40 ATP would be used to convert 10 molecules of glucose to pyruvate via glycolysis and then back to glucose via gluconeogenesis. Okay, so the correct answer is 40. This is the net ATP that would be involved in the process. Okay. Okay, so let's move on to the next question. Which of the following are true? Okay, A and C. Any more answers? Okay, so the correct answer is option C and D. Okay, so actually, option C, the animals cannot convert fatty acids to glucose because the glyoxylate cycle is not present in animals. Okay, so that's why they'll not be able to convert the acetyl coenzyme A that is derived from fatty acids to glucose pathway. Okay, both plants and microorganisms have that glyoxylate cycle, so they can convert the acetyl coenzyme A derived from fatty acids to glucose conversion. Okay, so this is the case with both C and D. So that's why these are the correct statements. Okay. Then glucogenic enzymes bypass four irreversible steps in the glycolysis pathway. This is due to reciprocal regulation. No, there will be regulation, but it is not reciprocal. Okay. Then glucogenic enzymes bypass four irreversible steps in the glycolysis pathway. This is due to reciprocal inhibition. No, this is also false. Okay, so the correct answer are option C and D. Okay, so any doubts in this question? Okay, if not, then are there any doubts from the week 7 of the course or from the PPT? Then we can discuss. Okay, if not, then we'll close the session for today. Thank you guys for joining in. Thank you.